It's my great pleasure to introduce Donna Pierre. Donna is the chair of Computational and Systems Biology program at the Sloan Kettering Institute and one of the leading researchers in this field. She is also on the organizing committee of the Human Cell Atlas project. Her lab develops machine learning methods for the analysis and interpretation of single-cell data. Donna has more awards than I could possibly remember. And, OK. Please take the stage. Thank you for accepting our invitation. It's a great pleasure to have you. Thanks.
OK. Thank you, everyone, for coming here. I know that this is largely a machine learning conference. And once upon a time, I did a PhD in machine learning. But now I'm a biologist. And machine learning is my toolbox. And we'll take some lessons in biology today. So one of the most fascinating questions is a really age-old question. So I want to address something that was asked by Leonardo da Vinci. How are we made? We start from a single fertilized egg, one single cell, and it creates the diversity of cells in our body. Our brain cell, this is NeurIPS. Our stomach cells are muscle cells. Every cell in our body are immune cells. They're all patterned and developed from a single cell that knows how to proliferate and choose new fates. And how does that happen? How does that process happen? One of the reasons that I got into computational biology from computational science is that cells are little tiny computers. They get input from their environment, signals from other cells, nutrients. They have all sorts of proteins that are their processing devices. They make decisions, they interact with each other, do their biochemistry and molecular biology, and decide whether they're going to proliferate, make more copies of themselves, differentiate, enter a new cell type, activate, release a molecule to talk with another cell. They're really like little computers, and we want to know how they work. And the remarkable thing is that all cells have the same genetic code, 3 billion letters that actually tells the cell. It's the blueprint of how to make our body, so how cells should differentiate and turn into the human body, as well as how each and every individual cell should function. The code for each individual computer and each individual cell type, they're all encoded with the same DNA code, which is accessible in a different way in different cell types. So different parts of this code are accessible in different cell parts. And this allows the cells to differentiate from one another and actually do very different things. We have hundreds and hundreds of cell types. And one of the game changers, technology always drives science, is single-cell genomics, and particularly high-throughput microfluidic-based single-cell genomics. What we have here is a microfluidic device, or a cartoon of it, where we have little droplets, microfluidic droplets that you could view as a tiny little test tube. And each droplet, it encapsulates a cell, and it could measure for every single gene in every single cell what gene that is and what cell it came from. And here we sort of see a little animation of how it works as the cells enter these tiny, tiny test tubes in order to get processed. And now that's been commercialized in a little machine that looks like a CD player. And so the data matrix, and this is just for one sample, and I run dozens of these a week, are 250 reads. Each read, you have to sort of associate what gene does that read refer to and what cell does that read refer to. Usually you get it at the end as a process gene by count matrix or cell by gene matrix. But the problem is that the original data is rife with errors and artifacts. And so this count matrix is certainly not very accurate. And the matrix is sparse. We don't get all the transcripts. We only capture about 5% of the transcripts, 5% of what each individual is expressing. So you have to take this sparsity into account. Now, in the field, zero inflation has taken root, and it's wrong. Zero inflation assumes that basically you have a little zapper and you choose entries in your matrix and zero them out, and everything else is untouched. But this is actually uniform sampling. Sometimes this uniform sampling leads you to capture no gene and no transcript from this gene, and you get a zero. But every single entry of this matrix is affected. It's like a sanding where no value is at its full value. It's a sampling problem where you look at a ball, you scoop up 5% of the balls, and those are the transcripts which you got, and that should be modeled properly and not with zero inflation. The first problem is, okay, how do we really handle all this data? People like to visualize. We're humans. We like seeing things in 2D, maximum in 3D, so people started with the typical dimensionality reduction, PCA, and PCA failed this data type, and actually for a very long while we couldn't visualize this high-dimensional data. This is when we were still looking at 30-dimensional data of proteins, and people, basically looked at 500 two-dimensional biaxial plots, and then one of my grad students came to NeurIPS and heard a keynote in which t-SNE was presented. So following a keynote in NeurIPS, he ran back to the lab and said, I'm going to try t-SNE. And voila, it perfectly split apart the different cell types. Each different color is a different cell type, and now they all split apart as they should. And basically, phenotypic space is really embedded in very low dimensional. So while you might have a matrix of 20,000 genes by 100,000 cells, that matrix really does reside in a very, very low dimensional and very, very structured matrix. And t-SNE, as well as UMAP and a couple other nonlinear methods, really capture this quite well. The reason why we have this sort of manifold, this low-dimensional manifold, it's not an accurate mathematical manifold, but that's what the field started calling it, is because cell phenotypes are a highly regulated process. This is the same data now in 3D, and you could see these exquisite non-convex shape of immune cells. If you take different immune cells from different healthy individuals, you'll get very similar shapes. That's because very few regulators really drive what cells how they can behave. We have tremendous co-regulation and co-linearity between gene modules and gene programs, and a lot of feedback loops and interactions between all these genes, really limiting and constricting the phenotypes that our cell can be in. Of course, we still have challenges in visualization, crowding, scale, and I want anyone who wants to build better visualization for this data is highly non-uniform. Different regions of the data have up to 5 orders of difference in density. I've seen people try and do synthetic data with uniform densities. The real challenge is to handle this data with such different densities, 5 orders of magnitude, and that really trips up many of the algorithms. One of the most common ways to actually model the data beyond visualize it, and it's nice to look at data in 2 dimensions, but you actually want to sort of model it at the very least in a dimensional space, is using nearest neighbor graphs. Every node in the graph represents a cell, and we connect a cell to its nearest cell, its most similar cells. Of course, the devil's in the detail, and depending on the data and the probability distribution of that data and your biological question, that's how you define your similarity metric, that's how you define K, or how many cells you're going to collect each cell to. But the idea is, once you build this graph, where obviously each cell is only connected to a very small number of similar cells, then you can really stay in the manifold. You can do geodesic distances, where each tiny step inside this graph represents a transition between 2 very similar cells, and then longer distances, rather than cutting across in Euclidean space. You do as distances of walks in this graph, or how many edges away 2 cells are, and that's been really, really powerful in many applications. Generally speaking, this data, as I said, is extremely structured. So if you look at communities in this graph, basically using any social network community detection algorithm, anyone that can scale, what you get are cell types, cell states, clusters. So this graph is highly modular with cell states and cell densities that are really captured as sort of clicks in this graph, and that's pretty much the most widely used method to cluster such single cell data. And the nice thing is that these graphs are connected, so even distinct cell types still share connectivity between them, and that allows you to capture the transitions of how one cell type might transition to become another cell type. Of course, these transitions, as I said, are very sparse relative to the cell types where most of the density resides. Another real fun part of this data, I started in the bulk genomics world where, I had to try and do some voodoo using 100 or 200 samples and learn something on 10,000 entities, which, as some of my colleagues explained to me, is non-rigorous science. Again, the reason it works is biology has a lot of structure. It's not adversarial. Therefore, even when you have a little data, when the structure is strong, when the signal is strong, you still have a chance. But when you have data sets with 100,000 cells, 1 million cells, finally you can do awesome things. For example, if you treat each individual cell as an observation of this little computer, as an observation of how this computer functions, then we can actually assume that molecular influences create statistical dependencies in the data. And using even Bayesian networks, circa 90s, Heckerman, out of the box, an algorithm I published in my PhD really gets you a correct reconstruction with no prior knowledge of T-cell signaling networks. What's particularly great about this is now that we can collect 10,000 samples from a single diseased individual, we can do disease-specific regulatory networks, take an individual, for example, a cancer patient, and try and figure out what's wrong in the regulation of that particular cancer patient because we finally have the numbers using this microfluidic technology. But really the thing that made me fall in love with this data type and really touch no other data type is the synchrony in the data. If someone would take a bone marrow sample out of you, one, that would be extremely painful, but two, you'd have all your immune cells. You'd have early stem cells and progenitor cells that are able to make and generate all the different variety of immune cells in your body. So any given time, we're regenerating our entire immune system, and you get the entire gamut from very early to very mature immune cells of all types, flavors, and colors. And so we can pick that up and get this dynamics, get the entire process of development and regeneration from one sample. If we trigger a process, if, for example, we try and induce the epithelial to mesenchymal transition, one of the biggest phenotypic changes a cell can make, which occurs both in development and cancer, again, from a single time point, we can get the higher dynamics of the process. And because we have so many cells and because we can treat each cell as an individual observation, we can even reconstruct the regulatory dynamics. All this from a single sample. And so I'm going to dig in a little bit into this pseudo time and basically the idea of reconstructing development and inferring the order of cells, the order of progressions of transitions from a single time point, and again going back to the nonlinear nature of the data. This is B cell development, these green cells. These are very early B cells. These are very late B cells, and as you can see, the process is highly nonlinear. It's twists and turns. This is a two-dimensional plot of just two markers, and you can see that one of them goes up and down, and basically a lot of development is markers rising and falling as the cell progresses through its different stages. Of course, any high throughput data, you get a lot of data, and the price is, it is messy, it is noisy, it is ridden with artifacts, and it is not nice. So you cannot treat it as absolute values. You know that your data is really noisy. And therefore, the graph structure I showed you earlier works so well, where you're only looking at steps between very near cells, allowing you to capture this sort of red line right here underneath the noise of all these noisy cells that might look closer than others. So the field really wants to take these cells, take these disassociated cells and order them, and the field succeeded to do that very well. In fact, it's ridiculously easy. Even PCA works not bad. The hard part is actually to get a very accurate reconstruction. And if you really want to discover, as I said, you have differences in densities, and a lot of the biology happens in rare cell types. So in our first application a few years ago, we managed to identify a really rare cell types, 7 in 10,000 cells. that had completely different signaling, that behaved completely different. It was a checkpoint. This checkpoint wanted to check that the jumbling of the DNA, that the DNA recombination that occurred inside of it is okay. And now that we can reconstruct normal biology and understand that checkpoint and where it occurs, we could actually understand that pediatric acute lymphoblastic lymphoma is actually caused when this checkpoint goes awry. And this little tiny population is the source of ALL, and that wasn't known until we could actually find this tiny new cell population, this tiny new cell state, and its novel regulation from this data by ordering it so accurately that we could pick up 7 and 10,000 cells. The data is very structured. We can capture bifurcations, and here I give a plug for Manu Sethi, who's on the market this year, and he is awesome if he's interviewing at any of your institutes. And so really, the way you can get structure in this graph, the way you can get bifurcations, is just by lengths of walks, by using waypoints. Basically, the direct route between two cells, if they're along the same path, should be more or less the same as a route through some intermediate cell. By the way, this algorithm was conceived in UBC, walking through the woods right here in Vancouver. But if they're on a different branch, then the direct path will be very different than the indirect path. And if you have many waypoints and you look at all the discrepancies between all pairs of waypoints, spectral clustering gets you the full bifurcation of the graph just like that, because this data is so beautifully structured. Trying to go all the way back to the question that Leonardo da Vinci asked us, and now we can try and answer him. We have a set of disassociated cells, and we're now going to look at early mouse embryonic development. We want to order them on the manifold along their ordering of how they sort of bifurcate and develop into all these different cell types. And we want to know not just how they're ordered, but what they might do, what decision making is going on. We want to know for each of these cells in our manifold, what is their possible future cell types, what is their propensity and their probability to go to each cell type. And again, one of the beautiful things about this data is that once we understand it, don't really have to do much. We don't have to do really sophisticated computation. We just have to understand our data. And basically building a Markov chain out of this graph really pretty much recapitulates textbook development. We use our neighbor graph, and we use the fact that we can do pseudo-time ordering quite accurately to sort of order it. We make a very strong assumption that development goes forward and not backward. It does hold in normal developmental processes. It's broken in processes such as cancer, and that allows us to build a directed graph, which we can turn into a Markov chain, and then we can look at the extrema states. We can find the final cell fates, those that have very little edges going out of them, clean them up a bit, and what we have is an absorbing Markov chain. Once we have an absorbing Markov chain, life is easy, everything is closed form, and we can compute the probability of each cell in our manifold to reach each of the terminal states. And we see this incredible plasticity that goes down. This is a very efficient and quick closed form solution that gives us the probability of each cell reaching each fate. And we can do that for every cell, getting a sort of measure of plasticity, the entropy of that probability for all cells. Of course, I can describe an elegant algorithm, but the proof is in the pudding. Does it work? So we applied it to early mouse development and the development of the endoderm. The endoderm is the germ layer that makes all your internal organs, your gut, your kidney, your lungs, your pancreas, all the good stuff. And so basically the data really organized along dimensions. Here we have multiple time points. E means embryonic day, before it's born, and you could see the cells organized along temporal orders. Then they have a proximal distal spatial organization. These are going to become extraembryonic tissues, making yolk sac and placenta. And this is the gut tube itself, organized along from head to tail. And if we zoom into this gut tail, we see that this gut tube is really organized based on each of its precursor organs. So even though it's a smooth gut tube, even though we cannot morphologically see any difference between any two cells in this gut tube, they're already organized by order and into clusters that match the behavior of the precursor premodal organs that will bud out of this gut tube. We have the premodal lung, the premodal pancreas, and all those good things. And what we see is that cell fates, we could actually transcriptionally see where a cell is going a full day before we see any morphological indication of what that might be. Of course, this is just a pretty graph, and the idea is, again, to get it very accurate, to really dig in. So if we go into the early days, the first decisions of the cell, whether it's going to become an epiblast and actually pattern the body and become us, or it's going to become a primitive endoderm and become extra-embryonic tissue that will support the developing embryo. And by looking at, again, this idea that we can actually take each individual cell and use it as an observation and see how the regulators in it change, we can actually find out what is the regulators that drive this decision. And we see that when both FGR1 and FGR2 are both high, that is when it goes to primitive endoderm, which is validated by experiments. We can go one day further and see that even though the cells have made a very distinct decision and have gone very, very distant from one each other phenotypically, here we have entropy. So of course there's very high entropy right at the beginning before the cells are making this critical decision. Then there's low entropy. The cells pretty much have decided if they're going to become a body or supporting cell. And right here we see high entropy again, and that was a surprise, and our biological colleagues thought that was a mistake. But we did all the statistical analysis under the sun, and we were convinced in our analysis. So the biologists went in and saw that these cells are plastic, that cells that were destined to become our body, that were destined to become epiblast, somehow jump outside, go to the extra-embryonic layer and assume the identity of this extra-embryonic layer. And we have this tracing which says once upon a time the cell was destined to be an epiblast, and now it changed its mind. And that was very surprising plasticity, and again, it was predicted computationally. It really, the biologist would never have guessed this under the sun. So given that we have such powerful technology and we can go so far in understanding biology, the community came together and decided to do the Human Cell Atlas to really comprehensively map all the cell types in our body, the transitions between them, and their spatial organization. And the idea of this consortium is to really go for scope, scale, quantity, and quality, with an emphasis on quality by working together. While most of the data currently being collected is still single-cell genomics, there are spatial technologies emerging, and this atlas is going to have both the single-cell genome-wide genomics as well as the spatial organization of these cells. And here are some emerging spatial technologies that will require a lot of computation in the near future and already beginning to emerge. So this community is already very big. It's a global open community. Anyone can join. You can see this web link right here if you want to join. And the data is going to be public. 10 billion cells for you to play with, your Candyland playground. And we're in need of more people to figure out how to analyze this complicated data. Already, a current-ish view. This current-ish view is a few months old, so of course these numbers have gone up. We've already collected very many millions of cells. This Human Cell Atlas is going to serve as a healthy reference for mapping disease. And we have many computational challenges here. For example, the scale. Most of the methods that we now work don't scale to millions and billions of cells. Data harmonization is a huge challenge, taking data from multiple samples, particularly from disease to normal. Almost all integration and harmonization methods sort of assume that cells are supposed to be similar. That's broken in disease, and these methods are horrible at distinguishing between biological artifact and biological difference. Annotating these cells, people spend hours just figuring out how to annotate these cells. That could be months of someone's time. factor analysis to get good gene programs, because these cells break down into gene programs. How this data factors between cells and genes is huge uncharted territory, as well as simply comparing disease to normal, using this idea that we can get regulatory mechanisms and much, much more. Of course, this is NeurIPS. So if I didn't show you guys, why is this not going? Oh. If I didn't show you guys some deep learning, I'd be booted off the stage. And this field has also done deep learning. There's quite a bit of interest in it, where you take this count matrix and put it into Latin space. It's been helpful in a lot of data normalization, data denoising, data integration. There's quite a few methods out there. This is a method by NeurIPS. Fabian Theis has a couple other methods, and this right now is a very active field. Again, these networks, these deep learning convolutional neural networks scale incredibly well. So it currently can deal with the most amount of data. It does have pretty good results, but we still are lacking interpretation of the Latin factors. And I actually have some reservations from these methods. And this is what's important about biology. Basically, our goal is not to predict, but to understand. We want to understand biology and the biological mechanisms. We want to understand disease, what causes disease, how to cure it. And the really hard thing about biology is often the outlier is the most important. The cells that really matter can be 1 in 10,000, as we saw with the ALL case. And really, machine learning is all about the common mechanism. Not about the outlier, and biology is about the outlier, and you can develop all these methods that are going to give you great ROC curves or any other metric that you want, and who will score amazingly, but will miss the important biology. So when you're doing computational biology, you have to keep the eye on the goal, and often the goal requires actually understanding something rare. I'm going to give you a concrete example with dendritic cells. These are certain immune cells. And the dendritic cells are the dark green cells in this plot right here. Who sees the dark green cells? There are a few, but you don't see them because they're rare. So together with our colleague, we actually enriched for them, and now we can see that these dark green cells, which you don't see here, have a lot of structure and a lot of heterogeneity in among them. Here we had only 2 of these cells. Now we have about, I think, 40,000 of these cells right here, and we see that they actually split into very rich phenotypic space. These cells are critical for immune response, they're critical for autoimmunity, they're critical for immunotherapy to work, and they're super rare. And they have a lot of structure. Also, cell types aren't necessarily clusters. That's why annotation is hard. And even clusters have further structure in them. This is sort of the topology of these clusters right here, and you can see that they're not uniform. We don't have a single hill here. And if you want to zoom in more, and the more you zoom in, the more you find structure of this data and structure of rare cell types. We can see that these metacells, these are sort of like real peaks in our density. Finally, when you zoom in again and again and again, they're defined by different gene programs and different gene-gene covariant structure. And covariant is actually one of the most important things in biology. When we studied the immune system of, that infiltrate tumors, when we looked at the critical regulatory cells that infiltrate tumors, we saw that cells that actually had very similar mean behavior of many gene cell types have very different covariant matrices. And one of the most important things that we discovered is that these covariant matrices, which are driven by the regulatory relations, by the gene-gene relationships that are really driving the biology, they're incredibly robust. Think about it, biology is molecular biology, it's biochemistry, it's stoichiometry. And while we ran the TILs and we compared TILs, or tumor-infiltrating immune cells, using 2 different technologies. One of them while I was still at Columbia, and the poor cells had to cross town, and our biggest factor, our biggest impact is taxi time or New York City traffic, all the way to when I moved to MSKCC and we could assay them similarly. Even genes and proteins, the covariate structure was incredibly robust across technologies, across artifacts. This is the biology in its essence. So a little, shameful plugging. I moved to MSKCC because I wanted to make a difference. And MSKCC is collecting an incredible amount of data, clinical data, electronic medical records, tens and thousands of pathology slides and radiology images, and the single cell technologies I'm telling you about now, we've already collected for hundreds of individual patients, and more is being collected. It's a data playground. And all of you are welcome to join and make a difference on patient lives, because we are looking for more people with your skills at MSKCC broadly, not just me myself. So I'm going to tell you a little bit about some of our games that we did in cancer at MSKCC. So acute myeloid leukemia is a cancer development gone awry. And at first you see each individual tumor is completely different. And but I know that it's not a processing area because the normal immune cells, they all overlap right here. And the cancer cells, we see a tiny population that overlaps right here. And these are really, really early progenitor cancer cells before they actually really go awry and go crazy. And so again, another plug for Elham, who's about to start faculty at Columbia in another month, and she's awesome. What we wanted to do is understand what happens exactly at that point that normal hematopoiesis breaks and cancer forms, and how does regulation change. And when you look at the classical methods, the healthy and disease are so different that they don't connect. All the normal methods can't really connect normal and disease. So again, we believe in covariation, and that's what drove us. We want to build a manifold, but driven by covariation rather than just normal distributions. We're based on a locally linear algorithm that was previously developed, but we're actually going to look for covariation between the components themselves. And this is wonderful because it's computationally efficient. We can compute the covariation in the much lower dimensional embedded space. It's much quicker. Covariation is wonderful except when you have to compute it. And it's also, again, represents the regulation that goes awry, the regulatory relationship that goes awry and then cascades into these global differences that we cannot reconcile. And when you run it this way, suddenly it all makes sense. The cancer breaks off exactly where it should, exactly in the development stage that it should be breaking off. And the cancer cells are organized based on pre-leukemic cells, and we can know that they're pre-leukemic still because they're not quite normal, but they don't have the driving mutation. And we can measure the mutation, and then we can see the progression, and we can see the covariation relations that change. And try and click up the page just to give you an idea. We can actually look at response to therapy. We have patients that get a bone marrow transplant and then relapse. And we want to understand what's the difference between responders and non-responders so that we can try and help the non-responders. And we have these dynamics where the responders, their tumor burden goes down, but the non-responders don't. And we want to understand what are the differences, what are the populations, what are the immune populations that differ between them. So we can use a hierarchical Gaussian process to get the dynamics, to identify the cell populations, and indeed we see some cell populations that really in the responders follow and raise up as the tumor burden is going down. And we see that in multiple responders, and they're just not doing anything. They're not growing in the non-responders. And again, these are very, very tiny rare populations, so we had to be very careful in our computation. And to try and understand why, again, we can go to regulation and integrate new data parts. So all that I talked about was gene expression, but we have epigenetic data. We have data that helps us indicate what transcription factors, what regulators are binding to the code, what are the potential regulators that can be regulating this. So again, using Bayesian inference, we can actually build a generative process. And we can say, okay, we have our observed data, we have all these latent variables of what are the different clusters, what are their binding patterns, what are their regulatory network and the covariation that it implies, what is the covariate nature. We build this into a full generative process about how each variable influences the others. Particularly, we know what factors as a prior can bind to what targets. And we use both this map of potential regulators to their targets, as well as covariation relations between regulator and targets to really predict the network. The ATAC-seq is only used as a prior. There are many important regulators that do not have binding motifs. And so we can have a data integration model. And we see, indeed, and predict all the regulators that differ between the responders and non-responders. Many known regulators, many exciting regulators that were just recently discovered, as many new ones that could define new therapies. Now, to really go quickly through a little bit of more cancer, just to give you a flavor of how complex this is, most of the cells in solid tumors are not cancer. Only about 10% are cancer, the rest is immune cells, the rest is a supportive tissue. And if you actually look at these cancer cells and study them and use different factor analysis, we see and can learn that the cancer actually hijacks early developmental processes. So the processes I showed you at the beginning for mouse development, those same process that creates the organ of the lung, the metastacists are using to effectively bud an organ, to bud a metastasis. So it's actually using the programs that the embryo knows to build the lungs to try and have a lung cell metastasize in some distant organ. This thing can actually suggest new therapies. So we want to understand how metastasis cells could survive in the leptomeningeal niche in the brain. These patients have 3 months of median survival time. We could identify that all cancers, both breast and lung metastasis, had the same 2 genes that created their ability both to survive there and to evade the immune system. And now, in less than a year, from data to clinical trial of a new therapeutic avenue. So this thing really has impact. Just to give you an idea of the complexity, again, cancer uses regenerative and developmental mechanisms. It sort of uses the healthy mechanisms that we have for its evil deeds. One mutation, one change in 6 billion base pairs can make it go completely different under injury. And this is how different this is. And you can see that in 48 hours, it's already behaving like an advanced pancreatic cancer, another one of the most deadly cancers. And the reason that this is, we learned, is because there's enormous crosstalk and remodeling between the immune system and the epithelial cancer cells. And the ability of the cancer and the immune system to talk with each other, and the cancer's tricking the immune system saying, I'm hurt, help me, can completely remodel the tissue within 48 hours. Therefore, spatial techniques are critically important. We have a rapid autopsy program where we can get multiple samples from the same patient. Here's some visuals of these. They're very, very different. They're very, very different in markers that people use to determine if immunotherapy is going to help or not. And depending on which of these samples we took, we might have made a very different decision about immunotherapy. Here's a zoom in, which I won't quite go into the details because it's quite towards the end. But these imaging, you can see how rich they are. There's a lot of information about how cells structure into neighborhoods, how they interact with each other. And we are really at loss as to how to interpret this data. Of course, we can try and segment the cells, and mask RCNN seems to work the best. This is this really crappy result comes from a tool that's sold to pathologists for $10,000 a user per year. So certainly we can do better than that and segment the cells. But even once we segmented the cells, and it's really hard and it requires a lot of training data and a lot of good training data, how do you interpret this? We're really missing methods. We did a quick other thing saying, okay, let's skip segmentation and just look at the data itself, at the distribution of the data itself. When we did it sort of in that naive way, we got something that at first looked cool, and then we saw that half our clusters were spatial artifacts. And then when we used covariation, my favorite tool, we could actually see that this actually clustered our cells according to known biology, resolved all the imaging artifacts because covariate relationships between genes and proteins are really the thing underlying biology. It worked both in the healthy lymph node, it worked in our tumor microenvironment, but even still, we're really missing methods to analyze, interpret, and find structure in this data. So again, a call to all of you. And in summary, I think this is an enormous field with enormous computational challenges, a lot of data that's going to be public. and a lot of need for everyone in this audience to get engaged, because we are behind in methods to use this data to really make an impact on biomedical health and cancer and in all diseases. So it can really, save lives. And this is wonderful members of my lab, current, past, and some collaborators that participated in the stories I told you here. Thank you.
Thank you very much. Are there any questions in the audience? There should be microphones. Go ahead.
Okay, so my question has two sub-questions. So one question is from computer science and the second one from biology. So from the computer science perspective, you have used the term manifold 20 times, but we haven't seen exactly where the notion of manifold, namely the existence of a neighborhood of the proper dimension around every point. So when you're talking about manifold, are you talking about a mathematical manifold or just about a space of reduced dimensions which captures correlations?
So I did say that earlier. A mathematical manifold can, be a decent approximation, but the field of single-cell genomics has sort of hijacked the term. What I can say is that the structure is itself much lower dimensional, in part because the data is so noisy and we can't remove the noise entirely. We don't know how accurate this manifold is. But dimensionality reduction really finds much lower dimensional structure in the data. When you sort of try and model it as a graph structure, you see very, very strong structure in the data. When you do diffusion maps, you see that most of the loading are on a small number of diffusion components. So it really is that this data, or the majority of the data, can be approximated by a much lower dimensional manifold. But it's not we don't really model it using an accurate manifold. I actually think that people coming to really try and use geometrical methods on this data can make a lot of progress in understanding this data, and that's currently lacking.
Okay, and the quick one about biology. So there are two topics which you didn't touch upon, if I'm right. So the first one is epigenetics, and the second one is isogenetic cells with different phenotype, which occurs typically when one is talking about cancer cells which may respond or not to drugs, and even though they are isogenetic. So do you take these two features into account, epigenetics and...
So again, I did a whirlwind description of many things. The single-cell RNA-seq data doesn't directly take epigenetics into account, even though we did integrate epigenetics data into the Symphony model, and we have a few models that take them into account. But looking at something like Palantir and a lot of our analysis, I differentiate between cell state based on RNA and cell fate. So you can imagine that there are many routes, many epigenetic routes to reach the same phenotypic state, and their epigenetics might be different. So if I take an individual cell, a single concrete individual cell, because of their epigenetics, I might be able to predict more what it might do had I known this epigenetics. But if I look at the population of cells that reach this state, and as a population predict, for example, in Palantir, the algorithm, given that I know that this cell reached this cell state, and taking into account the fractions of cells that reach these cell states and where they go, then the probabilities are correct for all cells in this state, but not a particular cell with a particular epigenetic state. But of course, now we're integrating the epigenetic data now that single-cell ATAC-seq is becoming commoditized. And that's new stuff ongoing in the field, which also has much room for even more methods development.
Hi, thanks for a great talk. I'm curious about how the methods used for, so the single cell RNA recording within one individual or a few individuals for a population of like a type of cell are analogous to things like evolutionary biology, epidemiology, and sort of the evolution of, or phylogeny, like if you can trace evolutionary developments in a similar way to the way you here have talked about tracing mutations in development?
So people are now using single-cell technologies within the context of evolution. So people actually, because single-cell technologies is based on sequencing, you don't need even a reference genome. There are entire communities, these evo-devo communities, which are just sequencing 100 different evolutionarily related animals and comparing them. In these comparisons, they're actually finding that actually the transcription factors that drive many of these cell fate choices really often do define a cell state, but the field of actually comparing development across organisms is extremely active, and again, could use a lot of methods development. In fact, pretty much any field, it's like a broken record. We need more methods.
Thank you for your talk. I had the following question. So basically you have a pipeline where you start with a tissue, you load it in device, you get data, and then you use this data you analyze it to prove or disprove certain hypotheses. So I was wondering, what if we could close this loop, and basically once we got the data, we could choose certain intervention, like what if we introduce, say, small interfering RNA to silence certain gene, and it will help us to prove or disprove the hypothesis, and basically have this closed loop similar to reinforcement learning.
That's actually the way that we work. That's one of the reasons I moved to MSKCC, because we do have collaborators, and we design the data together. I mean, again, we're really driven by biological questions. We have a biological question. We design the data we think can help us address this question. We analyze it together. A computational member of my lab working side by side almost every day with a biological domain expert, and there is a lot of follow-up, a lot of validation of predictions of new biology, a lot of design of new experiments as we discover things, and that's the real fun of working not with public data, but working with an experimental biological collaborator, and I'm really enjoying it, and I think the members of the lab are too. But of course, you need a biological partner.
Thank you.
Thank you for the talk. I had a question regarding, were you able to apply graph analytics in order to determine the de-differentiation process that leads to the cancer stem cells?
I don't quite understand the question.
Oh, sorry. So presumably an adult fully differentiated cell at some point de-differentiates into an early stage developmental cell that can then, as you showed, differentiate into a variety of different cancer cells.
Mm-hmm. So yes, that's one of the things that we study the most, how do cells de-differentiate. And again, some of the sort of pseudo-time methods alone, based on RNA alone, can't be entirely trusted, because they do assume that cells are going from a more immature to a mature cell. And we're developing a couple methods, one that almost made the cut for this, and I realized I had too much material, where we're using additional data, this case a TAC-seq, to try and orient the dynamics of where cells goes, basically assuming that if I can reconstruct the regulators and regulatory networking, I can see what regulators are active in a cell. I can see where those regulators might take the cell next, and that can help us direct the edges on the graph using other assumptions other than immaturity or maturity. But basically, these graph algorithms are very strong. The actual structure of the data that they capture is very strong, and the key is, how do you find the different similarity metrics? How do you define order of progression, and if you could use other things, and the example I gave you, transcription factor potential to push a cell into a different state, you could orient the edges using other assumptions than just immature goes to mature, and that's what we're using for cancer now.
Great, thank you.